A very good morning to one and all. On behalf of St. John's Institute of Pharmacy and Research, I, Kajal Singh, a Masters of Pharmacy student in Quality Assurance, would like to welcome you to our 25th session of SCIENTIA. Before commencing the session, let's have a small video of our college campus. Now, I would like to commence with today's session, which is entitled Solid Dispersion. It's a pleasure for me to introduce our eminent speaker for today's session, Mr. Prashant Dalvi, sir. Mr. Prashant Dalvi has been working as an assistant professor in Department of Pharmaceutics at SGIPR since 2016. He has completed his B Pharm and M Pharm from University of Pune. He has 9 years of academic experience. He has published research and review articles in 7 national and 2 international journals. He is life member of Indian Pharmaceutical Association and Indian Society for Technical Institute, and is also a registered pharmacist with the Maharashtra State Pharmacy Council. With this brief introduction, now I request Sir to please take over the session.
Okay. Am I audible?
Yes, sir.
Okay. So thank you, Kajal, for your nice introduction. Before starting my discussions, I would like to thank our respected principal madam and Scientia team for giving me an opportunity to conduct this Scientia session. So let me share the screen. Is the screen visible?
Yes, sir.
Okay. So topic taken for the Scientia session is solid dispersion. strategy to enhance solubility and dissolution of poorly water soluble drug. Okay. So here, so outline of discussion is, am I audible?
Yes, sir.
Okay. Outline of discussion is, okay, first I will be focusing on present scenario, then biological classification system, that is BCS and DCS classification systems. Then I will be focusing on solid enhancement techniques. Then I will take you on solid dispersion, fundamental design of the solid dispersion. Then I will be focus on the most important solid dispersion technique, that is amorphous solid dispersion, and then I will be take you on the recent advancement in HME and SD techniques, that is hot melt extrusions and spray drying uh, solid dispersion techniques, and then after that, okay, we'll discuss the case study, okay, of the solid dispersion. So this is the outline of the presentations. So let's start with the first point, that is the present scenario of this solubility, viability, and newly discovered chemical moieties. Yes or no? If you see, in pharmaceutical industry, the most common problem statement in pharmaceutical industry is low viability of the drug molecules with their poor aqueous solubility. Okay. So the aqueous solubility is one of the important aspects in the formulations and the development of any oral solid dosage form. Drug must be dissolved in aqueous solubility. Drug must be dissolved in aqueous, the solution. Drug must be dissolved in gastric fluids, intestinal fluids, in order to show their maximum therapeutic and pharmacological actions. Okay, about 60 to 70% of these drugs, you can see the diagrams, then you will get just chart. You will get the idea about 60 to 70% drugs falls into the class second and the fourth and fails to reach into the markets because of their the low solubility or poor solubility problems. Yes or no? Okay. As the new advancement in the combinatorial chemistry and also in high throughput screens, number of these new chemical entities, okay, discovered, okay, day by day. But the majority of the drugs come under the class seconds and class fours, okay, and unable to reach into the market because of their solubility problem. So that's why this aqueous solubility of these new chemical entities and permeability across these biological membranes are the prerequisites for the effective absorptions and therapeutic actions of any oral solid dosage form. As I already mentioned, about 70 to 90% of these new chemical entity discovered, developed, okay, come under the class second and fourth, okay, that reported the poor aqueous solubility, okay, and that's why it is come under the class seconds and the fourths, okay. So this phenomenon of this poor water solubility, okay, it's due to the structural and functional identified during the drug discovery process. There are the n number of the methods that we can improve the aqueous solubility of that particular drugs. Yes or no? Different approaches are there. Okay, during the preclinical trials, we can modify their structural activity relationships and we try to improve the solubility of the drugs. But there are the certain limitations, okay, in the improvement of this aqueous solubility of this new chemical entity during this preclinical development stage. That is the most important requirement, that is lipophilic nature to bind the biological target, okay, biological membrane to cross the lipid membrane. Yes or no? That's why we cannot improve the solubility of that particular drugs new chemical entities during the preclinical stage. Yes or no? So the different approaches is must. We can modify we can formulate that particular new chemical entity into the different formulations, and then we can deliver it to the targeted site. Yes or no? So according to their, if you see, according to their molecular weights, log p value, lipophilicity, physical and chemical properties of these new chemical entities, basically classified into the two main things. First one is the brick dust and second one is a grease balls. Yes or no? Brick dust, that indicate the solid state limited solubility of these new compounds, while the grease balls that indicate the solvation limited solubility of these compounds. Yes or no? And all these are the newly chemical entities. Okay, if you see, the majority of the drugs are in class second and class 4. Yes or no? About 60 to 70% of these drugs. Okay, so the physicochemical properties of all these drugs determine the formulation approach. Okay, to deliver that particular new chemical entity to this targeted molecules, targeted site. Okay, then biological classification system (BCS). Okay, so here, as I already mentioned, okay, drugs according to their physiochemical properties, okay, aqueous solubility, and bioavailability of the drug molecules, it is classified into the four main groups: class first, class second, class third, and class four. Okay, the majority of these drug compounds are in class seconds and class fours. Yes, class 4 molecules. So these BCS classification systems gives us a scientific framework, okay, for classifying the drug substance new chemical entities based upon their aqueous solubility and permeability. Also, it benchmark in the regulations of these bioequivalence of oral products, and this BCS takes into the account three major factors, okay, that is dissolutions, solubility, and interstitial fluids. You can see here, okay, if you see in the class first, marketed product it is about 35%, okay, and candidate is only 5 to 10%. Yes or no? In class seconds, candidate is 60 to 70%, and marketed product is only 30%. Okay, so about 40 to 50% drugs unable to reach into the markets. Okay, in class third, marketed product is about 25%, candidate is 5 to 10%. Almost all the whatever the drug which is present over there, all drugs are marketed in this class third. If you see the class 4, the marketed product is about 10%, okay, and 10%, candidate is 10 to 20%. Here is also, okay, about 10 to 15% of the drugs fails to reach into the markets because of the viability problems. Yes or no? So the aqueous solubility of the drugs and viability, it is one of the important aspects in the formulations and development of any oral solid dosage form. Okay, and the majority of these drugs come new chemical entities are found into the class seconds and class fours. These are the some recently discovered FDA approved uh, class seconds and the class 4 drugs, new chemical entities. You can see here majority of the drugs here in class second and class 4s, and hardly one or two drugs in class first and the class thirds. Yes or no? So here there is a lot of scopes for the researchers, okay, can formulate the different doses form, okay, to deliver these drugs, okay, to their targeted site. Now, another classification systems, okay, which is given by the scientist Butler, okay, that is nothing but developability classification system, DCS, okay, and these classification systems mainly focused on the importance of the dose on intestinal solubility of biorelevant media, okay, biorelevant media. And in this DCS classification system, scientists used, okay, the value of 500 ml of volume of available gastrointestinal fluids. But it is in BCS classification, if you see, if you want to show, or if you want to determine the solubility of any drugs, the in BCS classifications, they used only the 250 ml of these gastric fluids. Maximum amount of the dose soluble in less than the 250 ml of these drugs. Okay, it is in BCS classifications, and in DCS it utilized the 500 ml. That is the amount of the maximum amount of the drugs or the dose of the drug which is present into the 500 mLs. Yes or no? This is a basic difference between these two systems, DCS and BCS classification systems. Okay, so this DCS accounts for the synergistic nature of the solubility and viability of these new chemical entities. And according to that scientist, okay, okay, these DCS classifications again leads to the two subdivision of the two compartment, that is class second A and class second B. Class second A indicates dissolutions limited solubility of these new compounds, and class second B that indicate solubility limited of newly discovered chemical moiety. Okay, so these are the two different classification system. BCS and BCKS gives you a clear cut idea about the presence of these the new chemical entities into the different groups or class. So here, as far as the formulations of the oral solid dosage form is concerned, solubility is not enough. If you want to have the better pharmacological and the therapeutic action, drug must be present in large amount at their targeted sites. Then and then it will show the better pharmacological and therapeutic actions, otherwise not. So that's why the solubility is not enough. Solubility is not enough. Amount of the drugs, it must be there, then it will shows the continuous absorptions, okay, otherwise not. Okay, so here you can see these are the solubility of the some compounds along with their maximum absorbable dose. Okay, the equation is there, using you can find out the maximum absorbable dose of any newly discovered chemical moiety, any drug molecules. Okay, drug which is present into the amorphous stage, definitely it will shows the more absorptions. Okay, but later on after the 4 to 6 hours, there may be chances of these recrystallizations, nucleus formations, and absorptions of the such drugs will delayed. Yes or no? So accordingly, you need to select the process for the formulations of that particular drug compounds, right? Then the techniques, the selection guided for the drug properties. Okay, so appropriate solubilization technique is must. Okay, to deliver the drug to their targeted site. So aqueous solubility, physicochemical properties, viability of the drugs. Okay, guided us for in the selections of this technique. If you see this chart, then you will get the clear-cut idea. If you plot the graph, okay, by taking the log p value on x-axis and compound aqueous solubility of the drug in mg per mLs, okay, you will get the such a type of this spectra. So what it indicate? It indicate if your drug compounds have the more solubility, okay, so you can formulate it into the crystalline forms. Okay, here you can see here you can formulate it into the crystalline forms because of the no solubility problem. But if your compounds have the slightly less solubility, then the absorptions or the viability of the drug, that particular drugs can be improved. Okay, uh, can be enhanced. Okay, if it is formulated into the nanocrystals. Okay, nanostructures also you can say. If your compound is having the poor water solubility, okay, that times you need to convert it into the it in amorphous form, and then you need to formulate the dosage form, okay, for the targeted actions. Okay, targeted actions, and you can see here there are some hardly compounds. Yes or no? It is there in these gray regions. Yes or no? Also, the and lipid based formulations, okay, that having the greater log p value, okay, and that the you can be formulated accordingly for the targeted actions, right? So it gives us a clear cut idea about the selections of this appropriate formulation technique. Now, then the possibilities to enhance the dissolutions. There are two possibilities, okay, through which we can enhance the dissolutions of the drugs. Either we can go for the size reductions to increase the surface area of the formulations, or another important approach to formulation approach. Yes or no? So reduction in particle size by the micronizations. Okay, when you subject the drug molecules, okay, for the micronizations, definitely the surface area of that particular drug molecule will increase. Okay, and you can accordingly formulate the dosage form. Okay, and second one is a formulation approach. There are the n number of the formulations approach available. Okay, accordingly you deliver the drugs. So these are the different solubility enhancement technology or approaches. Okay, here nanocrystals, high energy polymorphs are there, amorphous solid dispersions, okay, liquid formulations, then use of the co-solvents and the surfactants, okay, that is mixed co-solvents concept nowadays, okay, it is very popular. Use of the cyclodextrins, beta cyclodextrins, okay. So these are the different solubilizations enhancement techniques. So selections of all these techniques totally depends upon the physicochemical properties of this drug substance, dose of the drug molecules, and their product concepts. Accordingly, you can select these techniques for the solubility enhancements. Okay. So if you see the details, the solubility improvement or dissolution improvement techniques, okay, it is classified, okay, in two groups, that is chemical or physical nature of these drugs, and second one is the formulations. Formulations, again, three different types, that is milling and macronizations. Liquid formulations are theirs, and carriers are theirs. Carriers, you can use the different carriers, okay, for the in the liposomes, macroparticles, and solid dispersions, and that is our point of discussion, solid dispersions. In liquid formulations, lipid solutions are there. Self-emulsified drug delivery systems are also there. Yes or no? Okay, in lipid formulations that can easily cross these biological membranes. Micronization and milling, this is also one of the formulations approach are there. And physicochemical properties, salt formations, polymorph selections, and inclusions of the some ionized group, okay, into the parent structures, that will also help to improve the solubility of this new compound, right? Yeah. So solid dispersion. Okay, what is exactly this solid dispersions? Solid dispersion is nothing but, okay, dispersion of one or more drugs into the hydrophobic polymers, hydrophilic polymers. Okay, that is nothing but solid dispersions. Okay, in short, it is just the molecular mixing of the drug and excipients. Okay, instead of action, you can use the word carriers also. Yes or no? Mixing of the one or more drug, okay, with the carriers. Carrier, it may be a crystalline, it may be a amorphous, it may be a combination of the both, or it may be a combinations with the surfactants also. Yes or no? Depending upon the nature and types of these your formulations, physical chemical properties of the drugs. Yes or no? So accordingly, you can select the carriers. Okay. So in short, these solid dispersions, it is a mixture of these two different things, that is the hydrophobic drugs and hydrophilic matrix carriers, okay, carriers at molecular level, that is nothing but solid dispersions. There are the different types of the solid dispersions, okay, it is classified based upon the uh, polymer used. Yes or no? So first generation solid dispersion, here crystalline polymers are generally used. Yes or no? Urea, sugars, different types of the sugars are generally used. First generations crystalline polymers are generally used. Second generations, that is here polymeric, okay, or amorphous form of these polymers are generally used. And in third generations, polymers, surfactants, and combination of both polymers and surfactants are generally used. Yes or no? So these are the different the types of these solid dispersions according to the polymer used. Okay. So examples of these crystallines and polymeric carriers you can see here, as I already mentioned, the urea, sugars, dextrose, sucrose, lactose, these are the crystalline polymers, hydrophilic in natures, okay, and polymeric carriers in that PEGs, PVP, HPMC, PVC, cross povidons, all these are the polymeric carriers, okay, used to form the to use to deliver the drugs to their targeted site. Now, amorphous solid dispersions. Okay, nowadays these amorphous solid dispersions, okay, shows the more uh, uh, area for the uh, formulations of the different solid dosage form. Yes or no? Okay, here in these amorphous solid dispersions, okay, the drugs are homogeneously dispersed in the excipients carriers in amorphous states. Why only in amorphous states? Why not in others? As you all know that these amorphous molecules have high energy. That's why it shows the more solubility in given solvents. Also, enthalpy and entropy of these molecules are very less. Okay, that's why it shows the more solubility. Okay, so a amorphous form of this API enhance the solubility by lacking the crystalline lattice and having the inherently disordered arrangements of the atom into the molecules. That's why the solubility of this form of these dispersions are more compared to the other systems. If you see the design of these amorphous solid dispersions, okay, it is a the mixture of this API and polymers. But this crystalline API is uniformly distributed, okay, or molecularly mixed with these polymers, okay, polymers. So this crystalline is API is converted into this amorphous form, okay, using different process. It may be a HEM process, it may be a solid dispersion process, and all other different process. So you can see here it is a molecular dispersions of this, okay, molecular level dispersions, okay, polymer molecular level dispersions of these amorphous form of these drugs, okay, and that helps to improve the viability as well as the solubility of these poorly water soluble drugs. It helps to maintain the supersaturations and the stabilizations of the formulations about more than 4 hours. Also, it helps to maintain the physical and chemical stability of the preparations more than 2 year. It also provides the maximum drug loading to achieve the optimal dosing and robust the manufacturing the process. Yes or no? So in this way, you can form these amorphous dispersions. There are the different advances in the technology that are generally used for this amorphous dispersion that we will discuss later on. Yes or no? But in generally, okay, it is designed in a such a way. In a such a way. Now you can see here the polymers which is present crystals. Yes or no? This is API which is present in crystalline forms. Okay, these orderly arrangements of this atom into these molecules having the less free energy, it's somewhat difficult to break the bond, whatever the bond which is present in between these two atoms, and it somewhat increase the solubility. But when you mix it with these hydrophilic polymers, okay, the whatever the lattice is forms having the less uh, less energy, whatever the lattice is forms or atom which is present into these molecules, okay, it is disorderly arranged, okay, that's why it shows the more dissolutions, more solubility, more dissolutions, and more viability when you come in contact with this aqueous medium, okay, or acidic or intestinal fluids. Yes or no? So these crystals convert into these amorphous states and stabilized by only the polymer which is used in the solid dispersions. Polymer which is used in the solid dispersion. Sometimes you can add the surfactants also. It will definitely it will be increase the decrease the contact angles and then it will increase the weighting. Okay, definitely it helps to increase the dissolutions. Yes, so I have added the one points regarding that surfactants in recent advantage. That time you will get the clear cut idea about this. Yes or no? But combinations of these different polymer surfactants is must as far as the design of this any amorphous solid dispersion is concerned. Yes or no? So why only these amorphous solid dispersions? There are the n number of the different solid dispersion techniques are available. Yes or no? If you see these amorphous solid dispersions, it's decrease in crystallinity. Yes or no? This amorphous form of this material have high solubility due to these disordered structures of the atom into the molecules. Yes or no? That's why this amorphous solid dispersion. Also, it helps to reduce the drug particle size, okay, to the molecular level that increase the surface area of the drugs. It helps to reduce the or absence of these aggregations and agglomerations due to the presence of the polymer which is present into the form. Also, it helps to improve the wettability due to the intimate contact with the hydrophilic polymers. Hydrophilic polymers. Yes or no? Okay. So that's why the amorphous solid dispersions, not all. Definitely, there are the certain limitations of these amorphous solid dispersions. Here, I have not highlighted that. But if you see these amorphous solid dispersions, okay, after the 4 to 5 hours, instabilizations of the product is takes place. There may be chances of the recrystallizations, nuclear formations. These are the limitations, but that you can overcome by the additions of the other third agents. Okay, but that also we will discuss later on. Yes or no? Okay, but these are the some highlighting points, advantages as far as the amorphous solid dispersion is concerned. Then the objectives of this design of this amorphous solid dispersion, as I already mentioned, drugs are the mixed into the polymers at molecular levels. Yes or no? So uniformly the drug distributed into the polymers, the presence of the charge on the API and the charge on these polymers accordingly distributions is takes place. Okay, most of the drugs are the weak base, which is present into the class seconds and class 4 of these BCS classification systems. Okay, if you add into these acidic polymers, amorphous polymers, drug salts form, and these polymeric salts shows the better absorption. Just see these diagrams, then you will get the clear cut idea about whatever these polymeric salts of these drugs. Okay, it shows the maximum absorptions of the drugs, and that increase the viability, solubility, and then viability of these formulations. But free based, okay, it shows the very means it is very less absorptions, okay, very less, okay, and that's why, okay, it shows the uh, subclinical sub plasma level concentrations. Also crystalline salts that somewhat increase the viability of these drug formulations, okay, but it is less to that of these polymeric salts. These polymeric salts, okay, makes the drug available, okay, in large amount at their targeted sites and shows the better dissolutions and absorptions, okay, which is required, okay, for the better therapeutic and pharmacological actions. So there is a significant improvement of the solubility of the drugs when you convert it into the polymeric form, right? Polymeric forms, right? In these amorphous salts, okay, there is a no lattice energy to overcome. In amorphous form, enthalpy of the solutions, it's become very less. The solution solubility also, it significantly, sorry, saturating solubility is also significantly increases, okay, and availability of the drugs at their targeted sites, okay, in large amounts. That's why it shows the more viability, more pharmacologicals and therapeutic actions. So amorphous salts have significantly enhanced saturated solubility, viability of the formulations. Okay. As I already mentioned in last slides, dissolutions of the crystallines, okay, it's somewhat difficult, okay, to reach their targeted sites, okay, also maintain the concentration of the drug in therapeutic index, yes or no, okay. So that's why we need to convert it into these polymeric forms, salt form, yes or no, okay. Just see these diagrams, okay, it shows the three different peaks. This is the flat lines, yes or no, that is dissolution of these crystalline drugs, okay, almost it is very negligible or less, okay. Then if you convert it into these amorphous drugs, okay, but as I already mentioned, there are the certain limitations of these amorphous drugs. After the 4 to 6 hours, there may be chances of the recrystallization, precipitation, nucleation formations. So that's why they decrease the drug released, okay, dissolutions, and ultimately ability of the drugs at the targeted sites are also very less. Yes or no? So suddenly in initially 2 to 3 hours, it shows the peak level. Yes or no? Okay, drug concentration is too much. It is at the shows the in therapeutic in this the drug plasma concentrations, okay, and it shows the you can see there the it shows the peak level, yes or no. But after 4 to 6 hours, okay, due to the recrystallization instability of this amorphous form, okay, it the release of the drugs at the targeted size, presence of the drugs at the targeted site is decreases, and it shows the less viability, right? So but when you convert it into the, okay, amorphous drugs plus amorphous salt form, or again when you add the crystalline inhibitor in that, it helps to inhibit this recrystallization, nucleations and precipitations of that drug molecules, and it will shows the continuous drug release after 2 to 3 hours, okay, after 4 to 6 hours also, and it shows the maximum drug release at the targeted sites. That's why the solubility, dissolutions, and the viability of the drugs also increases. Okay, this graph, just see this. Okay, it shows the absorption windows for the dissolved API. Okay, and continuous drug release and absorptions. Okay, of the drugs. Now, what are the different techniques for the formations of this amorphous solid dispersion? N number of the techniques are available. Use of these beta cyclodextrins, hydroxy beta cyclodextrins, HEM, hot melt extrusion techniques, spray dryings, yes or no. Then fusion methods are there, supercritical flue technology are there. Then some solvent evaporation techniques are there, yes or no. Electrospinning techniques are there. So all these are the different techniques, okay, used for the preparations of the solid dispersions. But here, as far as this amorphous solid dispersion is concerned, here I have, it is prepared by using the two important techniques, and that I have added over here. That is spray drying, and second one is hot melt extrusion techniques. Recent advantages in this spray drying that are also try to cover, okay, here in this session. So the amorphous solid dispersions, which is prepared by the spray drying. So what is exactly the spray drying methods? That first we will discuss, and then I will take you on the advances in the spray drying techniques. So if you see, this is the most widely and commonly used, okay, methods for the preparation of the solid dispersions. Yes or no? Why? Because of the avoids of these phase separations, and it's able to form the formation of homogeneous system. Homogeneous system. Okay. No doubt it is a solvent based system. There are the certain limitations. Okay, certain limitations. If you see after the formations of this spray dried product, whatever the product is formed, there may be chances of the some solvent stressed solvent residue, okay, is present into the final products, yes or no. So these are certain limitations of the product which is formed using these spray drying techniques. Okay, as I already mentioned, it is a solvent based technology. Different types of the solvents are generally used, okay, for the preparations of these amorphous solid dispersions or solid dispersion. Class first solvents, okay, class second and class 3 solvents are generally used. Class first solvent, it is a carcinogens, some harmful for us, then harmful to the human being, environmental hazards. Class second solvents, it is non-genotoxics, animal carcinogens, limited in pharmacological products, yes or no. And class 3, it's somewhat low toxic potentials to humans, long-term toxicity or carcinogenicity, then less toxics in acute and short-term studies and in genotoxic studies. That's why this, if you see, if you compare this class 3 with other two, okay, it's somewhat the less toxics or compatibles for us, okay, for the different formulations. That's why these class thirds are preferred solvent for the preparations of this solid dispersion using this spray drying technique. So, what is the general method for the preparations? The drugs, okay, prepare the or dissolve the drugs and carrier into the common solvents. Then pass through the nozzles, okay, and atomized into the fine droplets, okay, fine droplets into this the chamber, okay, containing the hot gas. Yes or no? When you atomized these solutions into the that hot that chamber containing the hot gas, okay, so what is exactly the process over there? This hot gas penetrates these fine droplets, okay, these fine droplets, okay, entirely surrounded by this hot gas. The penetrations of the hot gas, okay, takes place, and penetrations followed by the evaporations of the solvents, whatever the solvent which is present over there, okay, it will get evaporates upon the penetrations of this hot gas, yes or no. And then fine solid dispersion particles will form, and we can collect it into the separators, okay, and then we can subject it for the different formulations, different formulation. Okay, so here you can see the atomizations of these spraying solutions into the chambers containing this hot gas, okay, there are the different zones are there, hotter regions is there, cooler regions is there. Accordingly, the intensity, accordingly you will get the materials, yes or no. Okay, so the spray droplets heat inside the spray droplets, penetrations of these fine droplets, okay, by the heat, followed by the evaporation of the solvent which is present over there, and then you will get the spray dried form of the particles, okay, solid dispersion, yes or no. And then you can, this particle you can subject for the different formulations. Most important limitations of this spray drying techniques is that it is a solvent based techniques. Number of the solvents, okay, need to used. Some are the carcinogenics and all of the different properties, yes or no. And there may be chances to remain the solvents remain inside the final products, and that leads to the some medical complications, yes or no. So there must be thoroughly investigates, okay, these products before subjecting for the formulation. Now, what are the different advantages in these spray dried techniques for the formulations of amorphous solid dispersion? Okay, so first technique, that is Suba techniques, is there. Second one is polymer combination in spray drying, and third one is addition of the surfactants in spray drying. These are the some advances in amorphous solid dispersions formulations via the spray drying. Okay, we will discuss, okay, all these techniques in spray drying. Suba techniques, okay, it is one of the patented techniques, okay, US based pharma company, yes or no. And the first product developed, okay, by using this technology by this US based company, that is formulation of itraconazole, yes or no. Itraconazole is formed, okay, using this. Now conventionally is also available, these itraconazoles, it is one of the antifungal drugs, yes or no. Conventionally are also available, yes or no. But if you see this conventional product, okay, having the some limitations, yes or no. Limitations, okay. So whatever the metabolites forms of these itraconazole, that is hydroxy, okay, itraconazoles, okay, having the some side effects, yes or no. Accumulations of these hydroxytraconazoles in different parts of the body, that leads to some medical complications, yes or no, medical complication. So such a type of these disadvantages, yes or no, and also incomplete absorptions of this, yes or no. Hardly the 50 to 60% drugs shows the absorptions, remaining are totally the wastage, yes or no. So in order to overcome these limitations, okay, the scientists developed this technology, formed the nano-sized itraconazoles, yes or no, and that nano-sized itraconazole shows the better absorptions, okay, in at the intestinal pH, at the intestinal site, yes or no. Large surface area of the intestine with the nano-sized itraconazole particle shows the better absorptions, okay, shows the greater bioavailability of the drugs, yes or no. Technique is very simple, okay. It is just mixing of these API and polymers. It may be a hydrophilic, lipophilic polymers you can select, yes or no. Mix, dissolve into these common solvents, then atomize into the fine droplets, yes or no. And same mechanism here, penetrations followed by the operations, okay, of the hot gas, okay, and then find nano-sized, okay, particle, okay, you can separate, you can collect, okay, into these cyclone separators, yes or no, cyclone separator. And then these nano-sized particles you can subject it for the different process, yes or no, that is drying, blending, milling, and then formulations, yes or no, different process. And then you will get the actual formulations, yes or no. Okay, so this is one of the patented technology developed by the US based company, okay, US based company. Itraconazoles, it is one of the good example of this, the formulation which is prepared by using these uh, techniques. Second advantage, advances in this technique is polymer combinations. Polymer combinations in spray drying. Okay, the stability of the formulation and super saturation is must, okay, to have the better pharmacological and therapeutic action, as I already mentioned. If you prepare the formulations using the if you prepare the formulations using the hydrophobic polymers, definitely it will shows the drug release for the 4 to 6 hours, but after the 4 to 6 hour what will happen? Okay, it will the drug release will minimized. Okay, ability of the drugs will also decreased, and it will shows the somewhat less into the dissolution, absorptions, and the viability of the drug formulations. Okay, so in that conditions you can add the some hydrophilic polymers carrier as a pore formers, and that will help, okay, to improve the viability of your actual formulations. Yes or no? Actual formulations. So pore former is must. Okay, the combinations of the hydrophobic and halophilic polymer is must. Definitely it will help to increase the viability of your formulations. Example here I have added, if you see, amphiphilic polymer solu plus, yes or no, and copovidons to improve the supersaturations of these griseofulvin antibacterial antiprotozoal drugs. Okay, and this amorphous solid dispersion was prepared by using these solid dispersion techniques. Yes or no? So different hydrophilic and hydrophobic polymers you can combine to improve the viability of our class second drugs. Class second drugs, low solubility, high viability. Yes or no? So supersaturation of the drug must, as I already clear with these points, yes or no, to the targeted sites, then it will show the better absorptions. Okay. Another examples, as I have added here, the synergies between the polymers HPMC, methacrylic acids copolymers. Okay, type A eudragids, methacrylic acids copolymer type B eudragids, S, improving the solubility of the griseofulvin class second drug using the solid dispersion techniques, using the spray dried technology. Yes, so amorphous solid dispersion which is prepared by using these spray drying techniques. Yes, sir. So this is the combinations of the polymers, okay, in spray drying technique for the formations of amorphous solid dispersions. Then another advances in this spray drying technique for the formation of amorphous solid dispersion, that is addition of surfactant. Addition of surfactants. Okay, here addition of surfactants, when you add the surfactant in the different polymers, it will increase the wettability of the formulations drugs, and that decrease the contact angles, okay, between these two. Yes or no? Okay, so it will help, the surfactant definitely helps to increase the viability, dissolution, solubility, and then viability. Right? Examples here I have mentioned spray dried dispersion of the BCS class 4 drugs apremilast, yes or no, were manufactured, okay, using the copovidone, okay, vitamin E TPGS. Apremilast is one of the important drugs used in different skin disorders and all other psoriasis type of these disorders. Yes or no? Okay, so it is manufactured using these two. Yes or no? Vitamin E TPGS, it is a the synthetic water soluble version of this vitamin E. Yes or no? Okay, toco mersols. Okay, so these two polymers you can use for this, and non-ionic surfactants 280, vitamin E TPGS. Then ionic surfactants such as SLS were investigated in the formulations of these in situ nanoparticles using these spray drying techniques. So different types of these polymers, surfactants used to improve the viability of these formulations. So these are the some recent advances into the spray drying technique for the formulations of amorphous solid dispersions. The next important techniques, that is melt extrusions method, is very important techniques. Melt extrusions for the formation of amorphous solid dispersions. Yes or no? Amorphous solid dispersions. It is not solvent based technology. Yes or no? It is solvent free technology. That's why, okay, it is most important techniques nowadays for the preparations of these amorphous solid dispersions. Okay, so concentration of the drug in dispersions, it is about 40% weight by weights. Yes or no? And this technique is mostly used in the preparations of diverse doses form in the pharmaceutical industry. That is, sustained release pellets can be prepared using these techniques. What is exactly this technique? Okay, first drugs and the polymers you need to mix, okay, form the powder blends, and then put it into these barrels, heat the barrels. What will happen? Whatever the powder blend which is present over there, it will get melt, okay, and it will form the homogeneous mass, and then homogenized mass, okay, then pass to the extruder, and then extrudate will forms, and these extrudates subject for the different process, that is drying, milling, and so different pallets will forms, okay, and these pallets you can subject for the compressions, or also you can enclose it into the capsules for the further processing, right? So in this way you can prepare these dosage forms, yes or no? So this is the diagram, the diagram of these hot melt extrusions. You can see here the drugs, polymers, and the plasticizers also you can use. Okay, the mixture of all these things into this hopper. Okay, this is the extruders, rotating extruders. Okay, mixing of these all these three takes place inside these barrels. Yes or no? Okay, also blend formations, powder blend formations, and melting, okay, will take place only into the barrels. Homogenized mass will form, okay, and then these amorphous solid dispersions will produce. Amorphous solid dispersion is produced, okay. While preparing these amorphous solid dispersions, you can use the number of, okay, different types of these polymers, combination of the polymers, addition of the surfactant, plasticizers, okay. You can add all these different things and prepare the amorphous solid dispersion. These are the recent advances into these HME techniques, hot melt extrusions technology. Yes or no? So, what are the advances in this amorphous solid dispersion formulation approach? So, first advances is polymeric combinations in HME, polymeric combinations. Okay, and second one is addition of the surfactants. Yes or no? Addition of, okay, surfactants. So here in these polymeric combinations, as I already mentioned, the better stabilization is must. Okay, for and prolonged supersaturation is highly essential for the desires and sufficient drug released. Yes or no? Okay, generally, when you add these hydrophobic polymers with poorly water soluble drugs, that can leads to the prolonged release and extend the supersaturations, okay, in two different ways: intermolecular hydrogen bondings and hydrophilic interactions between the API and the polymers. API and the polymers. In this way, you can. But conversely, if you see addition of these hydrophilic polymers to the poorly water soluble drugs, that result into the rapid drug release, but after the uh, per, uh, 2 to 3 hours later stage, it can eventually lead to the nucleations and the precipitations. Yes or no? So that's why the combination of the polymer is must. Yes or no? In order to overcome the such a type of the limitations and the problems. Okay, hydrophilic polymers, hydrophobic polymers. Yes or no? Okay, and different types of the plasticizers. Okay, using that you can form these amorphous solid dispersions. Amorphous solid dispersions. Stabilization and the supersaturation is must upon the administrations. Another advances is addition of the surfactants. Okay, same to that of this spray drying technique. Here in hot melt extrusion technology, you can add the different types of the surfactant to improve the viability of our formulations. Yes or no? Viability of the formulations. When you add this surfactant into the formulations, that will help to increase the wettability and reduce the contact angle between, okay, contact angles, okay, to improve the solubility of the crystallines API. Yes, sir. For examples, amorphous solid dispersions of these apripitants using the soluplus HPC, PVP, okay, using hot melt extrusions you can prepare. And also, okay, the effect of the surfactants examine, okay, on these particular formulations on the API also. Yes or no? So here surfactants like vitamin E TPGS and poloxamers P407s in inhibiting the drug crystal growth in solid dispersions. Yes or no? These surfactants, poloxamer 40P407, it's inhibiting the drug crystal growth in the solid dispersions. Yes or no? Okay, which is takes place after the additions of the hydrophilic, the polymers. Yes or no? Polymers. Okay, so this is, okay, recent advances in this hot melt extrusion technology, right? Now, what are the different manufacturing process? Okay, and their recent advancement in that manufacturing technology. So here, kinetisols is one of the important techniques. 3D printing is there. 3D printing again in that selective laser sintering, fused depositions modeling, and direct powder extrusions and electrospinning. Okay, these are the recent advances in manufacturing techniques of these amorphous solid dispersions. Yes or no? Which is available, okay, nowadays into the markets. Yes or no? We'll discuss all these things. First is kinetisols. Yes or no? It is one of the important technique. Basically, it is inherited from this plastic industry into this pharmaceutical industry for the formulations of the different solid dispersions. Technique is very simple. Yes or no? Okay, here in this field, you can again add our drugs and different polymers, combination of the polymers, surfactants. Yes or no? Then it will enters into this. Okay, it will enters into this particular what you can say high speed rotating shaft. Yes or no? Process parameters you can adjust using the monitors. You see different uh, process parameters such as their temperatures, rotation scales, and all you can adjust using that. Okay, and due to the high speed of this rotating shaft, yes or no, whatever this blend, powder blend which is present here in the chamber, okay, it's get heated and a molten mass form. Yes or no? And the, the powder blend which is present into chamber, okay, it's get heated, but the temperature of that blends, okay, it should be greater than the glass transition temperatures and melting temperature of the drug molecules. Yes or no? This molten mass form in the inside the chambers, okay, it is discharged into the quenchers, and then the it's passed through these millers, and then you can collect the fine minute, okay, crystals into the collector. So this is one of the techniques, okay, kinetisols in use in the amorphous solid dispersions preparations, amorphous solid dispersions. Okay, then 3D printings are also there. So again, in that too, direct powder extrusions and fused depositions modeling. Yes or no? 3D printings. Okay. First one is direct powder extrusions. Just see these first diagrams. Here it is a single extruders are there. In that powder blends, you need to put and head these barrels. What will happen? It again the mixtures will get heated and molten mass is forms, and then directly it projected into the objects in 3D plates, 3D objects. Yes or no? That is direct powder extrusions. Yes or no? Second one is, if you see the fused depositions modeling is here. This is also one of the techniques used for the preparations of these powder blends, preparation of the amorphous solid powder dispersions. So these filaments, in these filaments you can put that mixture, heat these filaments. What will happens? The molten mass will forms and pass these molten mass to these, okay, mobile extruders, and then it will squenched on these mobile plates where our actual formulations, that is amorphous solid dispersion, will form. Yes or no? And third 3D printing technique, that is selective laser sintering. This is also one of the important. It consists of the different components, that is lasers are there, counter rotating powders, leveling rollers. Yes or no? And feed panels are also there. Yes or no? So here the laser projections decide the height of these powder blends. Yes or no? Okay, continuous bombardment of the lasers leads to the melting of these powder blends, and then it will be forms the it will quench on this plate, and then it will forms the printed doses form. Yes or no? Okay, so these are the three different techniques. Okay, that is kinetisols, 3D printing in the selective laser sintering, then these direct powder extrusions and fluid depositions. Electrospinning is also one of the important techniques. So it is a solvent-based technology. Here the drugs and the polymers you need to mix into the common solvents and then apply the electric currents across these needles and these collector plates. Yes or no? Collector plates. When you apply the current across these collector plates, whatever the metal which is present here in these plates, it will get on this plate, okay, and into these on these plates, and they form the nano-sized crystals on these collector plates. Yes or no? Then you can collect this and then subject it for the further processing. Okay, further processing. Electrospinnings are also important techniques. So these are the FDA approved drug products, okay, available into the markets. Yes or no, prepared by the different companies using different manufacturing techniques, using different polymers. Yes or no, their chemical names available into the different brands. Yes or no, nowadays. Products glance that also I have. Now here I have added one case studies. Okay, I will take only 5 to 7 minutes. Okay, case study. Then here meloxicam is one of the drugs which is insoluble into the waters. Okay, come under the class second drugs that shows the delayed absorptions and limited pharmacological actions. Yes or no? So to overcome the limitations of their solubility, supersaturations, and viability, okay, it is prepared into the amorphous solid dispersions to improve the solubility of meloxicam. Spray drying techniques basically used, okay, for the preparations of these meloxicams. Yes or no? So this is a techniques using you can prepare these solid dispersions, meloxicam solid dispersions, dissolve these meloxicams into the polymers in ethanol, stir it properly, adjust the pH using the NOH, and then subject is for the spray drying techniques. Accordingly, you will get the what you can say fine droplets, okay, and then you can process for the further doses form. So this is a, method. This is a formulations, different formulations prepared of these meloxicams, and it's showing the table showing the drugs and polymer ratio. Three different formulations are prepared. Okay, SD1, SD2, SD3. Meloxicam, the amount is sorry, concentration is remain constants. Okay, and this solid dispersion is prepared with the three different polymers. Okay, first plain meloxicams, then in combination with the HPMC. Okay, then HPMC with poloxamers 188s and this HPMC with the beta cyclodextrins. Yes or no? And it's studied for the difference, solubility, their particle size, and all other different X-ray diffractions properties. Solubility studies carried out, okay, in this way. Excess amount of these meloxicams, okay, which is present into the limited amount of the solvents. Shake it properly at 100 RPMs at 37 degrees Celsius for the 48 minutes. Subject it for the centrifugations at definite RPMs for the 10 minutes and then perform the UV analysis at the 360 degrees Celsius and accordingly find out the amount of the drug which is present into the solvent. Then accordingly after the solubility, particle size is also very important. You can see here SD1 showing the particle size in micron 10, SD2 is 9 microns and SD3. This SD2 it is in HPMC with the poloxamer 188. Yes or no? It's showing the less okay particle size. Definitely the surface variables and it will shows the more absorptions okay at this particular site, right? These are the water solubilities of these different formulations. SD1 shows the 3.187. Okay, HD2 it is a 15. Okay, solubility it is 15. Yes or no? Mg per ml and HD3 it is 4 mg per ml. So it indicate the particle size of the H2s are very less. Then the particle sorry solubility of the particle size of the ASD2 is very less. Solubility of the ASD2 is very high. So it will shows the better dissolutions. It will shows the better drug release and ultimately the viability of these formulations will helps. So the two different polymers are used, HPMC and poloxamers or beta cyclodistance are also shows the better results in combinations with that. So finally, these amorphous solid dispersions of this meloxicam was successfully formed using these spray drying techniques. Okay, using powder blends of HPMC and poloxamer 188 or beta cyclodistance also you can say exhibit the better solubility than free meloxicams and solid dispersions prepared by the HPMC hydroxymethyl cellulose alone. Okay, so in this way, these amorphous solid dispersions play an important role in enhancement of this enhancement of the solubility, dissolutions, and the viability of these drug formulations. Viability of the drug formulations. Okay, so with this, so thank you all for your patience listening. Thank you.
Thank you, sir. Now the session is open for questions. It's a great honor to propose a vote of thanks for today's session. I extend my gratitude to our eminent speaker, Mr. Prashant Dalvi, sir. In his relevant address on solid dispersion strategy to improve solubility, dissolution of poorly soluble drug, he has focused on present scenario of new chemical entities, type of poorly soluble drug, BCS and DCS classification, maximum absorbable dose, solubility enhancing techniques, fundamental design of solid dispersion, that is amorphous solid dispersion. Advances in amorphous solid dispersion formulation approaches and recent advancement. A sincere thank to Mr. Albert D'Souza sir for his encouragement and to all the other dignitaries of the management. A special thanks to our principal Dr. Savita Tauro ma'am for her immense support. I would also like to thank the coordinator for their guidance. Thank you for being there with us. Feedback link will be provided in your respective WhatsApp group. Kindly fill it. That will help us to